MTT assay का जो next step है, that is the solubilization. As we were talking about the conversion of tetrazolium into formazan compound or formazan product after this reduction step, this formazan is insoluble product. It gets accumulated in the cell around the cell surface and in the culture medium as well. Before we proceed to detect the color change of the cell culture medium, that is the intensity of the purple color, we need to solubilize this formazan product, because this formazan product is going to give you the reflection of that purple color, that is the light purple, dark purple, or somewhere in between the that light and dark scale. So if this solubilization is not done properly, the results may have some error, and that error will not reflect the true result of our experiment. For that purpose of solubilization, variety of methods have been developed, and those methods we can use depending upon our own need. अब इसमें जो चीज़ें देखने वाली हैं कि सिर्फ solubilization is a factor that needs to be observed, या फिर कुछ और भी factors हैं that needs to be observed so that the accuracy can be maintained. जो methods develop किए गए हैं उनमें ना सिर्फ solubilization के हवाले से जो है वो focus किया गया है, but it has also been observed or it has also been ensured कि इस process में जो color है उसकी stability भी maintain की जाए. Rate of evaporation, because 96 well है, 96 well में जो हमारा medium की volume बहुत कम होती है, so if the processes are time taking, then the evaporation process can affect the volume of the cell culture medium. So rate of evaporation को जो है control करना है to avoid the evaporation. Then अगर हम एक और कलर को सामने रखें, फिनोल रेड कलर विच इज़ पार्ट ऑफ द सेल कल्चर मीडियम, उसकी कितनी इंटरफेरेंस हो सकती है while developing another color in the cell culture medium. These methods include the use of acidified isopropanol or DMSO or dimethylformamide, SDS, or sometimes the combination of different detergent and organic solvent. Most of the time we in the lab use the acidified isopropanol to do the solubilization of this formazan product. Acidification gives us dual benefit. Number one, it is going to help in solubilization. Number two, it is going to convert the color of that phenol red from red towards yellow. The yellow color is going to have less interference when we are going to measure the color intensity of MTT assay as compared to the phenol red color. The pH of the solubilization solution is adjustable so that we can provide maximum absorbance, the opportunity of maximum absorbance. Otherwise, if we are not adjusting that pH, the absorbance can be interfered and the results may have some false reporting. The amount of signal which is generated depends upon several factors. While doing the MTT assay, we need to have an eye on these factors so as to ensure that we should get the actual picture of the cell viability. For example, the concentration of MTT assay, the length of incubation period, and the number of viable cells and their metabolic activity. The purpose of this assay is to determine the number of viable cells. So the concentration of MTT assay is a factor that needs to be observed. अगर हमारे पास cell viable cell, we imagine the viable cells are much more, but the substrate that needs to be metabolized by those viable cells, उसकी quantity ही कम है. In very first part of the assay, the MTT assay or the substrate has been metabolized, then there is no further substrate is available, but metabolically active cells are available, so the concentration matters a lot. Then length of incubation period, this is just like that. आपने एक एंजाइम एक सबस्ट्रेट लिया है, उनको आपने 5 मिनट्स के लिए इंक्यूबेट किया है, उनको आपने 30 मिनट्स के लिए इंक्यूबेट किया है. द मोर टाइम यू गिव, द मोर प्रोडक्ट विल बी प्रोड्यूस्ड. देन दैट इज गोइंग टू give you a color intensity different from the different time slots. This conversion of MTT to formazan by cells in culture is time dependent. We may see that one experiment, one assay was performed for 5 minutes, for 10 minutes, for 30 minutes, for 1 hour, for 2 hours. Let's suppose that our shortest time span for the incubation was 30 minutes as compared to 4 hours. If we look at this curve or this picture, we have the results from 4 different incubation periods, that is 30 minutes, 1 hour, 2 hours and 4 hours incubation. If we incubate this setup for 30 minutes, the optical density which we will read will be somewhere around 0.1, and if the same setup is being incubated for 4 hours, the optical density value will be read as 1.4. So this is just like an time dependent reaction, that the more time you give, the more substrate is being converted into product, and that product will give the color intensity as compared to the short time incubations. So we have to optimize the time required for incubation of color intensity development, so that the true picture should be presented for the viability of the cells. And I would suggest कि जब भी आप इस ऐसे को शेड्यूल करें, इस ऐसे को प्लान करें, आपका जो इंक्यूबेशन टाइम है वो सिमिलर होना चाहिए, अदरवाइज आपकी जो ऑप्टिकल डेंसिटी की वैल्यूज़ आएंगी वो कदरे मुख्तलिफ हो सकती हैं, एंड वी मे से दैट द रिजल्ट्स आर नॉट लाइक गेटिंग रिप्रोड्यूस्ड.